Good evening to all. Before starting our today's session, I would like to give some few instructions. Everyone is requested to mute their microphones and off their camera. Please do not click on present now option anytime during the webinar. For better voice quality, use earphones. Any query, please put it in the chat box. All the questions will be taken at the end of the session. If you have any connectivity problem, then we are going live in Facebook and YouTube also. Links will be put in the chat box. Now I would like to start the session. I, Dr. Janita, take this opportunity on behalf of Legal Savior Group to welcome you all on second day of Forensic Marathon web series. I am overwhelmed to inform you that Mr. Anurag Agrawal, Mr. Divesh Jayandani, and Mr. Sagar Jadhav, the founders of Legal Savior Group, have taken this initiative to bring awareness and academic excellence. It all started with an object to bring awareness about law amongst the people at large. This time, Team Legal Savior has taken initiative for celebrating Forensic Marathon of 14 days by conducting national as well as international webinars, which will impart amazing knowledge through various scientific aspects. Now, without taking much time, I would like to introduce our honorable speaker. So today the topic of our session is forensic genetics and human identification, which will be taken by Dr. Shiva Shankaran. He is molecular geneticist for Gene Explore Diagnostic and Research Center Private Limited. Sir has done doctorate from Gujarat University, worked in national and international lab of repute like CCMB, Center of Cell and Molecular Biology, and Nebraska Medical Center, USA, William Beaumont Medical Institute, Detroit. Awarded international distinguished awards from ARBO, ASHE, HS Foundation, and etc. Visiting faculty and mentor in 4 national universities. Have more than 13 design and IPR for credits in microarray and NGs. He founded Gene Explore Diagnostic and Research Private Limited and 3 other companies. PI and co-PI in 8 national and international research projects in genetics. Now I would request Dr. Shiva Shankaran to take over this session. Over to you, sir.
Hello, good evening, everyone. Uh, Thank you for such an elaborate introduction. And I'm also thankful to Dr. Shanmugam, Mr. Yadav for organizing such a good platform. Although I'm not sure how many of members are clearly visible because it looks like talking to yourself into the mirror. But anyways, I assume that everyone is part of it and everyone is listening to the discussion that we are going to have today. So I was given a task of introducing the fundamentals of genetics in forensic science. And I've tried to put up into a small presentation, which I'll go through in next 45 to 50 minutes. And I hope there are some intriguing questions that you will be having during my presentation and post my presentation. So I would like to have them at the end of or in between. If someone wants to have some particular query on any of the slides, please feel free to put on the chat room so I can see it on the chat room, or possibly there would be a raise a hand option anywhere here. So I would like to take questions over there also. So let me just get connected to the presentation that will help me and you to go through the session. And okay, fine, everyone. I hope everyone is able to see the front slide and listen to my voice. Assuming this is happening thoroughly and without any error, I'm going ahead with my discussion today that's on the fundamentals of forensic genetics. So to start with, The discussion I've divided this discussion into four major part. One is the first one will be basics of genetics and the terminologies, because I was said that there will be a heterogeneous group who some of them belong to the biology background and some may not belong to the genetics and are still very much novice in the genetics. So I started with very basics of genetics, what the terminologies, what the content of the further discussion here is, and the tissues of forensic genetics, because one of the major queries and one of the major purpose of this discussion would be, okay, what is the tissues that are applicable, what are the tissues which are good for forensic genetics. And I have taken a few examples over there. I'll be going through that part, and then I would be talking about the basics and the advances of forensic genetics, because I'm not much comfortable going through the history. But then, of course, we'll be looking for what is happening right now and what are the things that we are expecting in near future. And these advances are going to be very tremendous in deciding the course of forensic genetics, its acceptance in the legal framework. And lastly, I would be touching upon a very slight part of data interpretation. What are the different softwares available for data interpretation, and what are the pros and cons of these softwares that which are being used, and how that would be utilized to or used to present our reports, and how it can be defended and questioned in the legal framework. So, going to the first part of the genetist, the role of genetist. Basically, it's a very wide spectrum of people who are going to involve when it comes to a forensic part. There will be a crime scene. There will be people who are around the crime scene, collecting samples, collecting the, analyzing the crime scene, and there will be people who will be going into the legal part of it. And what would be a genetist doing into it? So this isn't a very brief role of it works. A sample either recovered by directly from the crime scene goes to the genetist, or through a forensic biologist who processes and samples and packages it and sends to a forensic genetist. So either way, it comes to the lab, wherein the lab person, the genetist, actually based on the availability of the instrumentation and based on the tissue forms, goes through some analysis on it, and he first analyzes the sample and then analyzes the victim or the reference samples, and based on the informations available from both the ends, he might go to report the content, or may add value add to the intelligence of whatever the information is available about that case. So what I'm trying to say here is not that every genetic report is going to be conclusive in terms of legal framework. So it will definitely value add. So we'll have to understand that not all this information is coming from a genetic lab or any forensic lab is going to finalize or is going to be finally acceptable from the legal framework. What we as a lab people are going to do or only value add to the current knowledge of the case. And it would be always in the terms of the legal claimant as well as the protestants, who are going to both talk about it and kind of analyze it, its acceptability in that particular case, and come to a conclusion in that. So we as in lab people are supposed to present our data in the more acceptable form and more palatable form to the present to the court. Coming to the part of basic genetics, like I'm sure at least this part of genetics everyone know what is DNA, the structure of double helix, the classical double helix structure, and the paired stirred form of it. And we all know that it's basically deoxyribonucleic acid, which is made up of four fundamental nitrogen bases, that is adenine, thymine, guanine, cytosine. And it's constant across the life forms. You take the smallest unicellular or the bacterial to the most advanced eukaryotes to humans. The basic nitrogen bases are the same, the ATGC, and there's no change in that. And this ATGC, the permutation combination, the long stretch of this four base pairs actually makes the DNA, and this DNA then transcribes into different functions of the life form. So, and in nature, if we talk about in the biological system, this DNA is never in naked form as it is shown here, the double helix structure. It is always associated into a protective protein association. Thus, the DNA is enveloped around certain proteins, wrapped around the histone proteins, specifically in the eukaryotes I'm talking about. And this eukaryotes makes this kind of structure, chromatin structure, which again further is enveloped in a particular organized in a particular fashion across the solenoid structure, and then finally forming a chromosome. This is the chromosome structure here. So DNA nakedly is very fragile in the biological system. There's a lot of proteins, there are a lot of fluids which are present in the biological system which might break it. So naturally, to have a protective layer, this DNA is always enwrapped around a certain number of proteins, which uh, evidently protects the DNA against any damage. And further, to add another layer of protection, this in eukaryotic system, this all structure, the chromatin structures, all the chromatin structures are enveloped into a double membrane structure that we call as nucleus, right? So how this bases are read. So this is the classical chromatogram, or the example how we humans can read this. So there is of course a instrument, and then there is a process to that instrument, which finally leads to this kind of peaks. And this peaks, we read it into G, C, C, T, G, A, and so on. And this is the language of God. So this is the genomic architecture defines what kind of aftermath it will bring about. It will bring about a protein, or it will bring about what kind of different physiological functions, and so on and so forth. In depends upon which life form it is present. But in general, all this genetic code, always running in anti-parallel of this DNA, are divided into triplets, like as it is shown in this figure. So every triplet consigns or confines to a particular amino acid, and this stretch of amino acid then coming together forms a protein. And this protein, interacting with several other proteins present in the cell, brings about a metabolic response. This metabolic response unitedly of that particular tissue results into a physiology. So you can see the complex algorithm and how all of them are connected to each other. So these are the fundamentals how DNA is connected to the complete physiology and the nature of the physiology. So this is how generally it looks like, where a DNA is transcribed into an RNA. RNA gets translated into a protein, and this protein then plays its function in the cell and in the metabolism. But sometimes there are some variations. This stretch of ATGC is variated at certain points, at certain sequence, and this will bring about a change, a differential RNA. And this differential RNA can bring about a variant form of a protein. So now here is the change that we are looking in. And if we are trying to see if everyone had the same genomic architecture and the same sequence, there will be no person to person, no individual to individual change. But we know every human is unique in his form, in his look, in his nature, and in his physiology. So this small variations are brought about by the small changes in this sequence. And this small changes is what we are looking for. Being a forensic genetist, what we are trying to see is how two DNA from two different individuals are different from each other. Classically, you would have heard about it and you would have studied a lot that 99.99% of genome, all the humans are same. That says 99.99, not 100. So 0.001% of variation is present from one individual to another individual, third individual. And this 0.001% of change is what we are going to analyze. We are going to see, and how this change can be so specific that it can identify an individual among crores and crores of another human forms in the globe. So DNA fingerprinting is the name which we have given to identify an individual based on the sequence that he carry in his body, in the each and every cell and each and every nucleus of his body. So this play, or this process of identifying the variation, is called as DNA fingerprinting, or DNA profiling, or human identification. And this is also used to identify the kinship, or it is also used to identify the paternity, maternity, patriarchy, and also ethnic group. And you can appreciate the smallest difference of this sequence is bringing about such a varied form of applications in science and in human forms. So this is what this genetic DNA and the sequence of DNA bring is the importance of it in science. So just going back again, how this DNA is architectured into solenoid form, to the chromosome form, to the nucleus, and within one single cell. And unitedly, this chromosomes, there are like 46 chromosomes in every human across the globe. If he's a human, he's gonna have 46 chromosomes, which says 23 pairs, 22 pairs of autosome, and one pair of sex chromosome. And this one pair of sex chromosome, the XX, or the X, one X and one Y will determine whether he's a male or a female in human form. And the rest of all these 23 pairs here, they are the autosomes. And this functional part of the genome, I will say, I'll come to it, what why I call the functional form. The genes which encode to protein are scattered across, and we have roughly 20,000, now more than 30,000 protein which are identified, are scattered, and their functions are relatively described in this small pie chart. So you can see almost only 3% of the entire genome is functional, and the 97% is still not known for what it is doing, what function it has. So we have almost roughly 3 billion base pairs. You can see the size. These 3 billion base pairs are encrypted into the DNA, and which is stored in a small nucleus within the cell. So this cell, this nucleus is nothing but our vault, our safe vault within our home. And this vault has the entire information of our human being the life form. So when I say the functional part, here is the functional part. You can see of 300 3,200 MB, the 3.2 GB part, only this much part, 2,200 MB is functional, which are actual genes or gene related to the sequence, which transcribe to a very small part that is just 48 MB into genes, and the rest of the supportive UTRs, the gene fragments, the pseudogenes, and so on. And coming to the right hand side of this, the intergenic DNA, which do not actually contribute to the functional part, but then they are present in our genome, which are mostly junk DNA, which is known as, or which does not transcribe into actual protein. But then this part is also equally important. Not later than 2 or 3 decades before this were not at all in of any use to human knowledge. But now we know there are a lot of important function of this part of non-transcribing gene, which we call, or the intergenic DNA, which is of the major part of human genome. And in this, for forensic application, we are talking about this interpressed repeats. When I say repeats, they are actual repeats of short sequences. It could be ATGC, 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 ACG, 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 and so on. So this short tandem repeats, what we call STRs, and the VNTRs, variable number tandem repeats, are of utilizations for human identification or DNA profiling or DNA fingerprinting. So coming to the second part of my discussion, what I was talking, what are the samples which are good for genetic profiling? Now, the simple one-line answer is any sample which contains DNA is good for genetic profiling. It's absolutely understood. If I want to do a DNA profile, I need DNA. So any sample that contains DNA. Now, the good question comes is which part of the human tissues contain DNA? Almost every tissue contains DNA. Either it may be blood, semen, hair, bone, tooth, epithelial cell, saliva, fluids, vomits, anything is going to contain DNA. But what would be the quality and quantity of DNA is the major question. So, in perspective of forensic science, from the crime scene, we normally get this kind of samples. That is the blood, the semen, the hair, the bone, the tooth, epithelial cells, sometimes scrapped, sometimes laid over, sometimes shed off skins. This is all what we get. Burnt skin, degraded, decaying tissues, and this is all what we get from the crime scene. And the reference samples are the best ones, the preserved ones, that is on the good shape, the blood, the buccal swab, pulled hairs, normally which are easily available, so that we can take it out from a person which is concerned and isolate DNA and keep it for the referencing. So these are the different tissue types which are available in the body, and different cell types which are available in the body. And you can see in all these tissue types, you can see small dots within the tissue. These are the nuclei, and these are the nuclei which contains the DNA. So you can appreciate one part is all this tissue forms have DNA, but the quantity of DNA would be different, excepting these two forms where the sperm and the ovum, the gametes of humans, in the male we have sperms, in the female we have eggs. They are haploid in number, or they have half the genome, and this rest of all tissues in our body will have a diploid genome, which will have 2n number of, so it will have two copies of everything that they have. And here we will have n copy, that is why we call them as a haploid cells, and this all are called the diploid cells. uh, a question if asked, which of them is good, both of them is good, both of them contain DNA, we can analyze both as and when required. Uh, but yes, of course, diploids are good because they get both the halves of the maternal and the paternal content in them. So naturally, the diploid cells would be better for any genetic analysis. And the most accessible, as you know, is the blood cell. So sorry, back. Yeah, this part. So we are not concerned about this part. Of course, yeah, leave alone the mitochondria. But then the cellular part is not of that concern, but the nucleus, which contains all this chromatin and the DNA, is of importance. So if you go through the epithelial cells of the sperms, this nucleus is well evident, and this nucleus actually harbors the entire DNA. And this DNA is of our concern and interest. Of all the sample types, I've taken few examples what kind of sample types we do get and what errors do we make in sometimes collecting the sample. So one of the classical example and the most frequently asked is hair. Is any part of hair good for the genetic study? Frankly, if you go back and study the anatomy of the hair structure, so only the partial, the proximal, the shaft part of hair is visible to us, while this major, the body part and the follicle part is embedded in the tissue in the skin. If the question is put of where the genetic content is there, the genetic content normally is residing here in the follicle part, which contains the bulb and the cells, the number of cells which are present here. This part of the hair, the shaft part, is nothing but a pure protein, a protein which is produced by these cells. So naturally, if someone brings up and cut hair and gives us for a genetic analysis, so that will be of no use because we will not have any DNA from this part. So the DNA that we'll be having is from this part. So if at all any sample is to be brought for a genetic study, the plucked hair is good because the plucked hair will have the follicle. Or if it can be visually seen, there is a bulb at the end of the base of the hair, it is good for a genetic study. So a cut hair will not be of any use, only the bulb or the plucked hair will be of some use. Similarly, of a tooth, I'm sure there will be some forensic odontology student here who are well versed with the structure and what I'm in the picture I'm showing. From a genetic perspective, this part and this part is of not much use for genetic study, but the rest of them is of use for the genetic study. So the enamel definitely will not have anything of genetic content. It's just an coating, which is a superior coating for the protection of the tooth, while the lower part, the tooth, the root, the bone, and the this cavity part is good for any kind of genetic study because those will have some cells, the blood vessels. This blood vessels will have some part of nucleated cells, and this nucleated cell is going to be having our genetic content preserved in them. So naturally, we need to have either entire tooth, then we can have to and scrap it and use for genetic study, or if there is a part of tooth, then we'll have to see which part of the tooth and how good the quality preservation of that tooth is there. Similar, one of the another commonest sample which are preserved, which is away from the crime scene is the bone. Not all part of bone is good, but then we know one thing is the bone is well supplied from of blood streams, and that's why we do get a good quality. But of course, the outer scraps are not of any use, the spongy part, and only the inner part, the medullary cavity, as well as the cellular part and the orifices are good for genetic study. But then it always gives a challenge. I remember when we were doing our PhD program, there was a sample of almost 5,000 years old, which was brought from Anthropological Survey of India. And it was a huge task of collecting DNA from that because after 5,000 years of weather eradication and chemical eradication, there was hardly anything left. But still, we got some amount of DNA, and we were the first group who sequenced the mitochondria from it, and it was like published long back. So naturally, it depends how the tissue has been preserved and what grade of insult or chemical, physical, and atmospheric insult has been done to the tissue will determine what quality of DNA will be availing from that particular tissue. So collection, preserving, transportation, and processing of the samples play a very determination role in genetic study or in availing the quality of DNA from that tissue. So nothing. So every part of this complete chain has to play their important role. It's not only the genetic lab which is going to play a very important role, or the collection samples, or the crime scene investigators are going to play important role. So everyone have to optimally understand the process, accordingly collect the sample, not only collect the sample, but preserve the sample. By the time it reaches to the lab, it should be at least well preserved. So nothing, so no loss of DNA is being happening during this transportation time. So assuming these are the few examples of what kind of tissues are brought or subjected to the lab. Assuming all these biological samples come to in, there are some genetic analysis done. Based on this genetic analysis and the purpose required, it can be the family study we can do the ethnic group study, we can do the individual study, whether how different and similar this individual is, and we can also identify between the two person whether it's the person in query or it's the person for the reference or the victim. So the likelihood ratios of this particular differences and similarities is measured, and this likelihood ratios actually gives us the information of how similar or different this sample is, or these two samples are. So coming to the technology part, we know one thing is the whatever information available and whatever the process optimization has been done so far is based on the Human Genome Project, which was the ambitious project from US NIH, NIE, sorry, and it started in around in 1990s and was completed successfully in 2003. And during this course of time, many labs contributed to the information, and it was one global project where many labs differentially took some part of the genome and they sequenced bit by bit, and finally the human genome was architectured and it was presented put for public use in 2003. And during the process, of course, the technology also improvised. By the time it started in 90s and by the time it completed in 2003, there was a huge difference in the technology, which actually helped the entire Human Genome Project. And the benefit of the Human Genome Project, the technological support and advancement was so dynamic that I'll give a simple example. The first human genome sequencing, it took almost 6 to 8 years and cost was almost 1 billion. 1 billion rupee or US dollars were spent on sequencing one genome. But if that project would have started instead of in 2000 and 1990, it would have started in 2003. By the time it completed, it would have just taken 3 to 4 months, and with a small cost of 30 million to 50 million, it would have been completed. And assume, hypothetically, if the project would have started in 2013, just 10 years from the completion, as it is being done now, the project would have taken only 2 days to complete and with a cost of only 4,000. Now you can see the difference from 2003 to 2013. In 10 years, the cost of sequencing has reduced down from 1 billion to 4,000. How important it is for forensic genomics. If there would have been a suspect which can be strongly brought under law and could have been punished based on the genetic evidences, the cost of proving it in 2000 or before 2000 would have been roughly this much. And today we can do it in a cost of less than this much. Of course, we're not going to sequence everything, but then this information was required. So naturally, so the technology advancement has helped us a lot. And this is a classical example of NIH, which states that how from 2001 to 2015, the cost has moved from 100 million to as low as 1,000 US dollars. So this course has been changed due to the technological advancements, and thanks to this technological as well as the database advancement, that we can now think of applying this knowledge of human genome and the genomic knowledge to the application of forensic and as well as criminology. The many technologies during this course of time which has been parallelly evolved, like qPCR, Sanger sequencing, microarray, array CGH, CGH plus SNP, the whole genome sequencing methods, the RNA-seq methods, which today I'll be also touching upon as the advanced and the futuristic approach of forensic study, and the mRNAs. This all are different technologies which in the last 2 decades have come into light, and all their role important process submission in the form of understanding and the application utilization for genomics. That could be forensic genomics or medical genomics or population genomics, but then yes, they are contributing equally and tremendously. Today, the comprehensive approach we have is from DNA to RNA to protein, which I showed in earlier slide also, to metabolites and the phenotype. And mostly in forensic application, we have DNA to phenotype. We are actually not much looking up to this three central part. But yes, in sooner coming years, these three central parts will also contribute significantly for forensic understanding. They are now doing a lot, contributing a lot to medical as well as health profession, but not in the genomics. But I'm seeing it not later than few years, RNA, protein, and metabolites from the crime scene also will lead to the forensic investigation, and definitely they will change the view, they change the prospects of what we are seeing forensic lab in coming years. And as this says, this genes to transcripts to proteins to metabolites, it will be a cut through pie, not a layered pie, layered cake, which will help us understand the entire case, and which will definitely change the methodology that we look into the processing samples as well as the collection of samples, because metabolites are something completely different and unknown to the forensic science and now, but then soon it will be also a very important factor to the forensic science. Coming to the normal course, the third part of section, how the process, the basics and the advance of genetics is done. So these are the three pillars of uh, the forensic system, forensic lab system. So the legal enforcement part, where the police agencies, the investigators, the detectives, they come into play. The scientific analysis in the laboratory part, and the geneticists, including the analyst and everyone, come into part. And then there would be the court system, the legal proceedings, and the prosecution, which plays their role. So these three are now connected with a very minute bridges, and in case-to-case basis, but soon they all will integrate into one system, and they will there will be a lot of crosstalk between this system to this, the enforcement to a scientific, and the legal proceeding to the scientific part. And once this is done, I'm sure the process of the judgment will be far easier and far easier and faster. Initially was only PCR and RFLP dependent, which then resulted into kind of STRs. And once the STRs came into play, the NGS came into play, and with NGS and the data, by 2009, DNA was important part of criminal justice system, globally accepted, and has also played a front role in sample collection and sample processing. Oh, how this DNA profiling helps, what are the basics of DNA profiling? Now we know each individual is unique in some small context of genome, and it is inherited from one generation to another generation. They're connected, familiarly connected. So it's not a standalone system. We are looking into a family when we are talking about one individual. And these subsets of genetic variations, which we call in terms of STRs and VNTRs, these are very clearly individually differentiated between one individual, though he may be very closely associated to his family member. Still, there is a genetic variation. So, two siblings of a family will definitely have genetic variations. Twins will be also having certain degree of genetic variation, but only exception to this, which still geneticists are confused and are not being able to resolve the problem, is the monozygotic twins. When we call monozygotic twins, they are two individuals from the same genomic architecture. That will provide a big challenge to us, and we'll have to find another ways that I talked, like expression, protein, and metabolites, which will not be similar to that particular two monozygotic twins. DNA typing and performed efficiently and reproducible and is globally acceptable. And here we are looking there to those part of genome which are not transcribing. That is, we do not look into the genes and looking into the other part of the genetic genomic architecture. So these are the major steps of sample processing, where we obtain the sample in whatever form it is being collected. And the first and the most challenging part of the entire system is the extraction of good quality of DNA. So this is the most challenging part of the system, and then comes the flow of DNA quantification to PCR amplification of multiple STR markers, depending upon the purpose the STR markers are selected. It could be of any technology that we are doing. It may be Sanger or maybe PCR or NGS systems. And then finally comes the part of comparison with the data samples, with the database, or with the victims or suspects, whether this is the same DNA or it's from different sources. So this is normal flow chart that we follow in most of the genetic forensic genetic labs, where this sample is availed from the crime scene or from the, it's a part of the evidence where the samples are preserved and brought to the lab. Sample collection and the flow moves on from STR amplification, and the data set is being preserved. Now, based on the investigation, there will be some number of suspects or the victims which will be identified, and based on this identified suspects and victims, again the same process follows, and a query of this question or this sample is put to the known person and the profile comparison is done. If this q equals to k is results, the inclusions match, the report is discharged and it is taken to the further investigation. And if it does not match, again it is put to the database, again another suspect or another person sample is collected and it is again matched. Still the q equals to k doesn't come up. This process goes on cyclically, and not necessarily every time it is conclusive. Sometimes we may reach to a non-conclusive methods, non-conclusive results also. So whatever the kind of queries are, this is the process of step which is being followed. I've just shown this flow chart, but how it goes in the lab, it happens this way, where the sample is collected, it goes for a DNA isolation, it goes for amplification, PCR-based amplification, then put up into the Sanger or the kind of NGS, and these results are obtained. And these results are obtained in terms of number, and the person is put up into a numeric comparison, and this numeric comparison is compared to the database. And this is how the general flow of process happens in any genetic or in a forensic genetic lab. So based on the instrumentation, we can look into the entire process, whether it is STR, as I said, it's a short tandem repeats, or it's a BNTR, which is variable number tandem repeats, or we are looking into the mitochondrial part or the Y chromosomal part. Why I've separated these two parts is, we know more from the genetic perspective, when we talk mitochondria, it is always maternal lineage, and when we talk Y chromosome, it is always paternal lineage. We are comparing it with the familial aspect. If you are comparing two individuals, it is simply looking into this two part. So sometimes, in some cases, this information also becomes very vital to establish a connect between the crime and the victim and the suspects. So this, whatever it may be, STR, VNTR, mitochondrial, or Y chromosome, it could be PCR based, which is absolute now. Very few labs are performing about purely PCR based results. Mostly it is Sanger sequencing based, or few advanced labs are working it on NGS or next generation sequencing based results. So based on all these three technologies, the reports can be generated. But I'm again repeating, PCR based reports are now absolute or questionable in court of law. So naturally, these PCR based reports are not much reliable. They are further to be validated and confirmed through Sanger sequencing or by NGS. These two are the advanced technologies and are now acceptable globally across any nation, across any court of law. So comparing to these two technologies, that is, I'm taking C, that is Sanger capillary electrophoresis, Sanger sequencing, and the NGS. Here, there are advantages and disadvantages. The major advantage of C is it is an established technology accepted by every court, unquestionable. And the major disadvantage is the low throughput. The low throughput, and only select of one or two cases can be done at a time. And the NGS comes with the major advantage is it's a huge throughput, a very high throughput, and we can do at a time hundreds of cases together, or plenty of cases can be compared with each other. While the major drawback or the disadvantage is the cost. Mostly the NGS comes with a very high cost per sample. But yes, the data output is more. And of course, when we are talking about capillary, we are talking about only limited mixture of STRs. But here we can do both SNP STRs together, and we can greater level of multiplex capability is being permitted by NGS. So it depends on how important the case is and how soon the data information has to be availed to the legal justice, and depending upon how much budget allotted to that particular case is also important. And the selection of the choice of technology will depend on many factors, and the facility available in that particular lab also makes an important decision on choice of technology. So I'll just these are the basics. I'll go to a bit of advances in the forensic genetic, and next generation sequencing is the future. Although I don't see it a lot of forensic labs today using it, but I'm sure within a decade's time, most of the labs will transcribe or move on from Sanger sequencing or capillary electrophoresis to next generation sequencing. And the flow becomes very simple, more robust, and more reliable. And huge amount of data is available at a single go. So it's again, the process remains sample collection to quantification, library preparation to sequencing and data analysis. And it's more cost effective when we do a large number of data, large number of samples in that, and it gives us, as I said, power of multiplexing a greater number of STRs. So as examples, this is common for both capillary as well as HGS, but the PowerPlex fusion gives us with the 23 autosomal STRs, 3 YSTR, and standard amylogenin, which can help you to identify between two individuals and very clear identification. And the power index can be of 2.3 into 10 raised to minus 32. And this is a classical example of how two data sets look alike and how the samples of A, B, C, D, and E can be compared with each other. And don't worry a lot about how sharp these peaks are, how broad these peaks are. There is a very established data software which puts all these figures in together and gives you a power index. And based on the power index, you can clearly establish as the sample is comparable or non-comparable, whether they are biologically related or not, or these two samples from the same individual or not. So it will be the data set will be available. So all the choice of different powerplexing or different multiplexing will depend upon different systems that you select, and the choice of what is the purpose, whether you want to compare it with the database or you want to compare it with between two individuals. You can select any of these technologies which I've been talking. And based on the technologies selected, you can collect 5x primer or 5x master, or you can select different types of kits which are commercially available for comparing the data. Of course, the samples, as I said, I extracted DNA from any tissue type, or the FDA punches, or the swabs. They all can be completed in 85 minutes if it is an advanced genomic lab. And when I say 85, it's less than 2 hours. And that makes sometimes a very important, in especially the high influenced, high media captured rape cases. This becomes a very important time frame, 2 hours, and within 2 hours you are able to produce some report to the legal systems. I was talking about the fourth part of data interpretation. One of the most complicated part of of this forensic genetics is the data interpretation. But now, fortunately, we have a lot of approved and globally accepted database softwares, which are soft genetics, BITL, TSLV, and this come with their own benefits and loss. But then, depending upon your technology, depending upon your instruments, and depending on your budget, you can select. But the end point is all of these technologies, all of them give you liberty to perform forensic genetics in a very low amount of DNA. Here, there is an example of data analysis done from a sample which was only 500 picogram. When I say 500 picogram, pico is 10 raised to minus 12. So we come with gram, microgram, then comes the nanogram and the picogram. So from 500 picogram, it will be few cells. When we just have one hair follicle, which has few cells left in that attached to that hair follicle, still the data is available. Now, several times you might have come across that data was that in a good amount of genomic DNA was not available to conclude anything. In major number of cases which goes unsolved from the genetic lab, the major challenge or the one of the major problem is the quantity of DNA. Now, with this kind of advanced technologies, even with a 500 picogram genomic DNA, data can be retrieved, and you can see the data quality. The data quality is really good. We tried to go down also, and there were few cases. This is not from my lab; it's from another lab. They've done it from 62 picogram. So the earlier one was from 500 picogram. This is from 62 picogram, and still the data quality is appreciable, usable for a report generation of forensic genomic lab. So that's the quality. That's the technological advancement that is now is available, and it is a a bit of advanced lab can have this kind of system from very small lab as good as one single cell. From one single cell, the genomic DNA can be extracted and can be utilized for reporting. So this is an example of how this advanced technology can do or work on different type of population also. So we can use a higher multiplexing, and across the ethnic group also it can be studied from ditch to Himalayan to Pygmy samples, and still the data can be available. We don't have to go through several trials, errors and trials to go through different STR markers to incorporate the different diverse ethnic groups. So these are some of the examples of how NGS, STRs, SLRs, SNPs are beneficial across all the data loci. These are all the different data loci. 1338 being the most commonest, 1358 to almost even THO1. THO1 is again one of the classical example of how data analysis or how the STR loci can be selected. So these are again few examples. I will not go into detail of each and every data set which is being generated through these systems, but ideally you need to know, understand from this data, from these graphs and from these data sets how many loci has been incorporated. Now, if I want to simply, if you want to take up an example of two data set, which is a better data set, the number of 22, the loci, and how they are scattered in the genome. So how many YSTRs, how many mitochondrials, and how many ECC loci are available in the data is important. So from perspective of crime or from legal perspective, how many such loci makes the call more reliable or makes the report more reliable. So these are the few examples of comparison between NGS and CE. And of course, as I was talking STRs from autosomes, I was also saying and putting emphasis on mitochondrial control region. Nowadays, the mitochondria is playing a more important role in identifying humans or identifying two individuals. And this small data sets helps in identification of two individuals separately with more confidence. So the confidence level increases with putting up mitochondrial data into your STR data. So I will show you an example of how this helps in one of the reports, and how Y chromosome also helps in. So this is a classical example of why initially why STR markers were not much appreciated in data logging, but now we know why STR is also equally important, because there was an article which is hello, hello,
Yeah, sir, it's audible.
is audible.
You can go.
Right? That's
Yeah,
perfect.
it's audible. Yeah,
It's
yeah,
not disconnected. Fine. So this is a classical example of across male-female mixtures from Y chromosome to mitochondrial to autosome. The entire data set can, in a single course, in a single run, give us the information of that particular individual, and this can be put up to any national or global database, and it can be compared with any person right sitting in here in India to US, and the connect between these two person can be established. So these are the examples. Now, talking about the advances, what we are seeing in next decade, whatever I've talked was the past and the present. Now, what we are looking at, I will be talking a very small overview of what we are going to see in soon future, in near future. This is more comprehensive form, but the human DNA, but the microbial DNA and the RNA, because we know, okay, we are surrounded by microbiome, and specifically after this COVID and the global pandemic, we are now know okay, many more microbiome are also important to understand how we integrate with each other, and how this two integration can also help in forensic testing. Someday you will be seeing a report where not only the human genome but the microbiome of that particular locality was also compared to establish the connect between the victim and the uh, uh, kind of the claimant or the suspect. Of course, this is not available today, but yes, sometime soon. And how the RNA6 also, the RNA from that particular, what so far what we are talking, the STRs and the VNTRs were purely of genomic DNA. But now the RNAs are also equally important. Now, for example, which tissue, once it is isolated into DNA, we don't know which tissue it belongs to. But this RNA can help us which tissue was also present over there, whether it was a semen sample or a blood sample which was collected, and how much of semen sample, how much of blood sample can be also quantified based on the data from the RNA-seq technology that we are proposing to incorporate into forensic genetics sooner or later. So this is how it will work. It works in medical fraternity right now. So I can give you an overview of this, like how from the RNA, its library is prepared, it reaches to the sequencing, and how the algorithm sets alignment, and after the alignment, it can be compared with the database. And once this database is compared, we can say whether this tissue belongs to what kind of, or whether the sample belongs to what kind of tissue, and how it can be contributed to alligated person or the non-alligated person. These are some examples, and how these pipelines work is also, this is an example for your knowledge. Of course, time will say how more robust this particular algorithm can be utilized for forensic solutions. So I try to touch base from base to the most advances and the future, so that the participants who were listening to this get an idea of what they are into, and how dynamically this field is changing, and how dynamically the next future, the next aspirants of this particular field have to keep to be on the toes and keep learning every day, so they are kind of up to the mark for the challenges they will be put up in near future. So I think I've come to the conclusion of my discussion, and I am up to the point. There are a few 3 minutes, I guess, left for discussion. And if anyone wants, or if anyone is having any questions, please you can put up the chat room, or I guess Shanmugam or maybe Jadav can help me with questions. Shanmugam, how am I supposed to know what queries we have?
So still off, we ask them for the chat box for the
Okay,
questions.
so I'll just
No
open the chat
questions.
box.
We will wait for 5 minutes. So.
Okay. I guess that's a question here.
Yes, sir.
Okay, so it's from Anurag. If I'm reading it rightly, can DNA report be fabricated? Suppose a DNA report links to the person with an offense, but on the contrary, person suspected from the commission of an offense proves his alibi that at the time of offense he was not at the place of offense, but at some third place, which is in all matter challenging the veracity of the DNA report. See, when we are generating a DNA report, we do not claim that the DNA collected or the sample showing the DNA was at the very same point of the time where the crime was enacted. Now, let me very elaborate this in a simple language. कि DNA presence say कि the person was someday present there. We do not put a timestamp on that. No genetic report right now available put a timestamp that this person or this sample collected is from this particular day and this particular time. So the timestamp cannot be put up to genetic samples. So it can connect the person. If someone denies that I've never been in that place, we can prove him wrong because your DNA was present here. That means at some point of the time you were here, but we cannot put a timestamp on that. So the fabrication part will be always a question. Well, if you're asking the DNA fabrication in terms of reliability, no, there's no question of reliability. It's 100% sure that if the DNA matches with your DNA, the report matches, definitely your DNA was present. But if you're asking whether you can be connected based on this to the crime, it will depend upon the legal system to prove that whether that person was present at the time of crime in that place. I hope I've answered. If you can unmute Mr. Anurag, I may be able to understand what he was trying to ask. But meanwhile, I'll move to the other question. What will be the cost of human genome sequencing if we performed it today? I think one of my slide answers that it could be as low as $1,000. And if you're talking on Indian perspective, it will be as low as 70,000 to 80,000. 70 to 80,000, you can sequence your genome within a period of, say, 48 hours. Where is this place for NGS? I did not get the question. Who's this? Where is the place for NGS? It's right here in Ahmedabad, right in our lab. If you're asking, and many of lab now NGS are available. Next generation is not an American product which available in only in Europe and America. I think if you want me to answer this question in a different way, how many next generation sequencers are present in India? There are 34 to 40 sequencers that I know are present across length and breadth of India. In Chennai, I know at least three installations. I know in Chennai there are three NGS sequences present, to my knowledge. Okay, that is the ML gene can be extracted from and the ML gene is not referring to the enamel protein, not from the enamel of the dentine. If you are asking for, it is expressed in the sex chromosome. Can be extracted from enamel protein. No gene, no DNA can be extracted from a protein. It has to be from the cell, specifically the nucleated cell. So protein is different. Protein is a product of the particular cellular function. So we do not extract DNA from protein. We extract DNA from cell. And which cell? The nucleated cell. Say, for example, I'll give you an example for here, blood. So in blood, we have many different cells. Like we have RBC, we have platelets, we have nucleocytes, we have eosinophils, we have basophils, neutrophils, lymphocytes. So not all these cells have nucleus. The RBCs and platelets have no nucleus. So we have only the WBCs, the white blood cells, which are nucleated. So if you challenge me to isolate DNA from RBC, no one in world can do that. It will be always from the WBCs. So we should know which cell type we are talking about, which cell type we are using for extraction of DNA. And if you are trying to ask whether this gene is the same for the animal, that two are again different genes. Amelogenin gene is different, and which do not, I'm not sure it is involved in the animal formation of a dentine. I'm not sure about this part.
Yeah, thank you, sir. Sir, there is one question related to genetic disorders.
Yes.
Can the genetic disorders such as chromosomal abnormalities or single gene disorders can be useful in crime scene for identification of the victim?
Yeah.
While coming to that?
That's an interesting question. If I put in hypothetical situation where a person with a genetic disorder was suspect to be present in that particular crime scene, so that genetic disorder person DNA can be compared to the samples and we can still prove it. Even we don't need this high-end technology for that. As simple, for example, sickle cell. We know in sickle cell the RBCs are sickle shaped. So if we have a blood sample over there, which shows under a microscope a sickle cell, and the person we know, the suspect, is having a sickle cell anemia, we can connect him. Out of 100 people, we can now narrow down to at least one or two people, because not everyone is going to have a sickle cell. The frequency of sickle cell anemia in population is less than 0.01%. So when we are talking, the number is reduced down. The suspects are can be narrowed down to some very few numbers. So many such genetic disorders or chromosomal disorders can be attributed or can be utilized. So the genetist should be sharp enough to pick up the hints, and based on this hints, he can narrow down his complete flow, study flow, to from many number of suspects to one or two number of suspects.
Thank you, sir. So there is one more question. Do we can extract DNA from unmaturated RBC?
Unmaturated WBC, yes. If you're talking about from bone marrow, because when we say the erythropoiesis, the process of RBC formation, it is physiologically called as erythropoiesis. Erythropoiesis initially comes from nucleated cell, the premature cells, RBCs. So while the process of erythropoiesis, the denucleation takes place somewhere in second or third part of maturation. So before the denucleation takes place, before the nucleus is pushed out of the RBCs, yes, we can have, you can use those cells. We not clinically and legally proven. It's an hypothesis. It's an experiment still going on. There are R&Ds going on. It is said that when some person touches a surface or when we do anything, we leave traces. And of course, as part of touch, the first thing that comes in exposure is the skin, and we have dead skin which keeps on shedding off everywhere. And this touch DNA, yeah, this shed of skin and the cells can be used for DNA extraction and can be used for the genetic studies. But still, the reliability is not that great, not that great, because the shed of skin, the shed of cells are mostly keratinized cells which have very few or traces of nucleus. So not a great amount of DNA, not a great amount of quality of DNA is availed from this shed of skin. So naturally, we'll have to kind of still improvise our technologies from sample collection to sample process to the technologies like next generation sequencing that we use to study this. Once all this process are optimized and reproducible, we'll be able to kind of gain approval to the touch DNA concept also. But yes, the things are going in that direction also.
Thank you, sir. So one more question. Sample collected from the crime scene, if it gets contaminated, or if you have a mixed sample of 2 or 3 victims, can it be identified?
Yeah, it can be used for identification. It's not like we cannot use it. But yes, there are other levels of investigation, and there are different softwares which are available. Say, for example, it's we are getting a sample, a blood stain, and unfortunately, the blood stain is of both the victim and the suspect. It can clearly be established. But if we have more, DNA or DNA from more people in that, that also can be differentiated. But it all depends upon the capacity of the genetic lab and the experience of the genetic lab to differentiate between two, three, four, five individuals blood sample in that particular same very sample. It's not impossible, but it's of great difficulty to differentiate and distinguish different samples. But yes, it is doable. It's not impossible.
Thank you, sir.
Mm. Thank you so much, sir, for a wonderful session. Also, I would like to ask you to stop sharing your slides so that I can share my slide.
Okay, and how do I do that? Stop screen share. Okay, done that.
Okay. So, again, thank you so much, sir, for lightening us with the wonderful session. On behalf of whole legal Savior team, I would like to present you the certificate of appreciation. Although this is a virtual space, but please do accept this certificate, sir.
Thank you very much, Dr. Janita.
Thank you, sir. Now, since we know this is a forensic marathon going on, so every day there will be webinar. So tomorrow at same time there will be another webinar on role of forensic physics in identification of victims by Dr. Mukesh Sharma, who is Associate Director in Physics in Police Forensic Science Lab, Jaipur. So I hope that tomorrow every participants will come and attend the webinar. It's done, sir.
Hello.
Yes, sir.
Sir, can you hear me?
Yes, sir. Your voice is audible.
Shiva, sir. I think sir has left the meeting.
I think sir has left the meeting.
It's okay. So thank you all
So thank you all
participants.
participants.
Do join us for
Do
tomorrow's
join us
session.
for tomorrow's
Uh,
session.
the timing would be the same
Timing would
from.
be the same for.
So kindly all
So
the
kindly all
participants
the participants
give a rating. Thank
give
you.
a rating. Thank you.